Adrian Harris. I'm the director of the Waggoner Center for Alcohol and Addiction Research here at the University of Texas at Austin. I'm also a professor of neurobiology at UT Austin. Our center at UT Austin is to explore the biological mechanisms of alcoholism and drug addiction with the long-term goal of developing new medications for treatment of these diseases. We have about 20 investigators on campus that are part of the center, some from the Department of Chemistry. All of the investigators are interested in different aspects of addiction, some ranging from the very molecular, genetic aspects, very basic aspects of interaction of drugs with proteins and protein function, all the way to animal models of addiction and even human studies of addiction and of student drinking that's being done by clinical psychologists as part of the center. So we have a wide range of approaches to understanding alcohol use, alcohol actions, and drug addictions. One of our directions is the development of new treatments. And these would be new pharmacotherapies, new drug treatments. And these are based on our studies of finding targets of drug action, understanding how these drugs affect the brain in very basic, simple systems, and collaborating with medicinal chemists like Professor Steve Martin to devise new chemicals that will provide better treatments for addiction.
Hi, I'm Stephen Martin. I'm the M. June and J. Virgil Waggoner Chair in Chemistry and Biochemistry. I'm also affiliated with the Waggoner Center. One of the principal functions of the Waggoner Center is to try to develop, I think, long-range therapies for treating not only alcoholism but other forms of addiction. And one of the exciting things one of the exciting aspects of research that's taking place in that institute is work of Adrian Harris, who's trying to identify various proteins that are overexpressed in people who do have addiction to alcohol. And one of the reasons for doing that is to then find small molecules, probably peptides as the initial leads, that bind to these proteins, with the thinking that if you can identify small molecules like peptides that do bind to these proteins, then down the road one might be able to identify molecules that mimic these peptides and also bind to the same proteins. And so these other small molecules would be non-peptides that might then serve as potential drug leads for clinical treatment of alcoholism. It's a long way down the road. Adrian and I have only sort of talked in general terms about collaborations we might develop. We've recently written an NIH grant, and we're sort of waiting to see whether that gets funded, but it's an exciting prospect to try to develop therapeutic agents for treating not only alcoholism but other forms of addiction.